I'm going to talk about pain, mostly from the bladder, obviously, in the context of this conference, and neuroplasticity. So that's how sensory systems from the bladder change in both acute and chronic form. Affiliations, nothing, self-funded. I live about 6 metro stations from here, so it's not a big deal. I have a senior citizen metro card, so that's my funding. I've given $2 to the organization for them. Bladder pain. We're going to talk about transduction, very little, because there have been many aspects of this conference that have dealt with this. Yesterday I went to a discussion workshop which really dealt with this in great detail. But then, more importantly, we're talking about chronification of pain from the bladder and both the peripheral and the central components. So, bladder pain, transduction of bladder afferent signals. Now, you know, and as I said, this has been dealt with in great detail. I don't want to bore you with more things, but you know that the transduction of signals in the bladder doesn't necessarily all have to do with pain. It also has to do with reflexes, with micturition, with voiding, and the sensation of fullness. And we all have a range of sensations from the bladder, from mild fullness to more intense fullness, to unpleasant fullness, to pain. So it is a complex situation, which in the last decades have been shown to be much more complex than we thought. The primary afferents embedded into the bladder tissue are actually closely related to the urothelial cells to the point that the transduction mechanisms involve the urothelium. Until some time ago, we thought that the urothelium was just a barrier, an epithelial barrier, but now we know that, of course, it plays an important role in the transduction. And this is a very simplified model, but of course there are TRPV1 receptors and others in the urothelial cells. There is ATP release, there is purinergic receptors. So it's a complex environment in which there is a neural element, of course, the primary afferent, but also a non-neural element, which is the urothelium. And they both interrelate to produce signals, which may have to do with pain, but also with sensations, with non-painful sensations of fullness, and with bladder reflexes. We have looked in the last few years at this interaction in the peripheral end with the role of the cannabinoids, and particularly looking at the role of cannabinoids. These are the kind of sensory receptors you find in the bladder. These are single unit recordings from the mouse bladder. I think yesterday one of the scientists told me that it was difficult to do, so there you are, I just showing off. There are 2 populations of receptors in the bladder, as you know, a low threshold and a high threshold. The thresholds are distributed in a kind of more continuum way when you look at this, at least in the mouse. In other species, they are more separated. But anyway, there are 2 different kinds of afferents. These are probably concerned with the reflexes and the initiation of the non-painful sensations. The high threshold are more related to pain, to inflammatory conditions, and so on. We looked for the expression of cannabinoid receptors. We found that it's only CB1 receptors are expressed in the bladder, just in the same way as in the brain cortex. This is just for analogy. So the same kind of receptors that we find in the CNS are also found in the periphery. And this was an interesting thing because, as you know, cannabinoid receptors are found more and more outside the central nervous system, and that opens a new avenue for potential therapeutic targets. I don't know if you will see this in detail, but we did a lot of co-expression of cannabinoid receptors with substance P, with CGRP, with P2X3 receptors. I show you only for the sake of time, because we don't have much time, co-expression with TRPV1 receptors. TRPV1 receptors are expressed, as I showed you before, in the urothelium and also in the nerve fibers, in the afferent nerve fibers. And here you see in green the cannabinoid receptors, in red the TRPV1 receptors, and when they merge in yellow, you see, you may not see it very well, but anyway, there are fibers and the umbrella cell as well as the epithelium, which are double stained. So not stained, double fluorescent. So cannabinoid receptors of the CB1 type are expressed in nerves and in non-neural elements of the bladder. So what does that mean? When we looked, and we did a study with AstraZeneca with this compound, when we look, this is an agonist, it's a mixed CB1, CB2 agonist. When we looked at individual nerve recordings from the bladder, we found that agonist, CB1, CB2 agonists produce a reduction. And this is important. It's not an abolition, it's a reduction of the responses, a modulation. So it's a modulation factor rather than an inhibitory factor. When you wash out, this is all done on a in vitro preparation, explanted tissue. When you wash out, the afferent recovers. If you put an antagonist of this particular compound, then of course you have no effect. So essentially, the main point I wanted to make on this thing is that we have a complex signaling mechanism peripherally in the bladder, and that complex mechanism involves neural and non-neural elements, and that not only involves the more traditional TRPV1, P2X3 purinergic mechanism, but also involves what you make not so conventional transmitters and modulators such as cannabinoids. So what happens when we're talking about now a longer period of time, the kind of thing that will be more related to a clinical setting, chronification. Now, we've known for a long time, this is very old studies done in my lab by Jorge San, in this case it was from the ureter of guinea pigs, so it's not the bladder, but it's just the tissue close to the bladder, that we have the two populations, the low threshold, this is the distribution of pressure thresholds, the low threshold in green, the high threshold in red. And we know that when this population of high threshold afferents is subjected to some kind of insult, like in this particular case, we were reducing the oxygen content of the perfusate. This is also done an in vitro tissue preparation. When we produce a local anoxia, then this population becomes low threshold. So this is an example, an indication of peripheral sensitization. So we have known that for a long, long time. We also know, and this is again old stuff from the lab, that this complex environment in which the urinary system, this is again ureter, but ureter and bladder is involved, between the epithelium, the nerve fibers, and the muscle, is a microenvironment that can influence profoundly the entire setting in which these sensory fibers live. In this case, this is an in vivo study in rat using this experimental kidney stone model of Maria Delejan Berardino. So these animals essentially are injected with a bit of dental cement. They produce an artificial kidney stone and essentially have a renal colic. And you can see that the normal ureteric motility, this is before anything happens, is high frequency and low intensity. Once the ureter is trying to pass the stone, then this develops into in bursts of high contractions, which are probably the basis of the well-known clinical renal colic. This can last for days, but more interestingly, even after the stone has been expelled, and this is 4 days after the stone has been expelled, the abnormal motility with a high contractions that go way above the painful threshold is still there. So the microenvironment in which the afferents live is very important in the understanding of the chronification process of the bladder. And going back to cannabinoids, we use the cyclophosphamide model to produce sensitization. Here we have the stimulus response curve of the afferents in blue. After cyclophosphamide, of course, the afferents become much more active. You can see there is a sensitization. And when we use the CB1, CB2 compound, then interestingly, we produce, going back to normality, but only at a high pressure intensity. So essentially what happens is that it reduces the sensitization of these afferents to the high intensity stimulus, to the noxious stimulus. So at that point, we really wanted to, we knew that we hadn't seen CB2 receptors in the bladder, so it was probably a CB1 action. But still, we wanted to be sure, so we used two antagonists of the compounds, one for CB1 and one for CB2. When we block the CB1, then we see that the compound doesn't do anything. The afferents are still sensitized. When we block the CB2, then the CB1 action is still there. So essentially, the sensitization reversal is mediated by CB1 cannabinoid receptors. So you can see the complexity of the chronification process of pain involving traditional and not-so-traditional neurotransmitters and neuromodulators. Finally, a few words about central components, and this is something that I've been working for some time, and this is a kind of my Billy Graham message, sort of kind of a little bit of early morning arousal, making you think. Everybody knows, it's in the literature everywhere, that everything to do with the CNS is central sensitization. And these are the things taken. I've removed the authors so as to protect the guilty. Generator of pain hypersensitivity. You can see you can pick up any paper and everything is central sensitization. If you have an increased pain sensitivity, it has to be central sensitization. And this originates, the earliest I could trace was more than 100 years ago from James Mackenzie, who proposed. He was a British surgeon who proposed, in the case, actually for visceral pain and from organs such as this one, that when afferent signals come into the spinal cord, this is taken from his own words, this portion of the spinal cord may be looked upon as being rendered abnormally excitable in consequence of a violent stimulation from the organ V. So the idea is that if you have this enhanced activity from the periphery, you get enhanced activity in the CNS. And it is true that what I call evidence from the bottom up, when we do in the lab, is we look from basic processes and see how we can support that. We look at reflexes, neurons, pathways, and so on. The famous wind up that you may have heard of. So if we stimulate the periphery, yes, you get CNS neurons that become more and more excitable. But I mean, these are seconds, so we're talking about a very short-term action. We can look at neurons in the dorsal horn. That receptive field expands, both the peripheral and the activity from the bladder as well, the activity from the CNS, from a visceral organ. So there is some evidence that reflexes, neurons, and pathways become acutely more excitable with a peripheral input. And even intracellularly, we looked at intracellular signals, membrane traffic of receptors. And this is just a kind of a very quick trip through all of these, the millions of papers. We can see trafficking of glutamate receptors to the membrane of dorsal horn cells, for instance, within 10 minutes to 3 hours of, in this case, it's instillation of capsaicin into pelvic organs. And you can see how this happens. So there is a fair amount of evidence that in animal models that acute noxious stimuli, and this is important, generate transient increases, and this is important as well, on many CNS systems, motor, sensory, and autonomic. Now, this, unfortunately, has been taken as a kind of article of faith, and this produced a link between pain sensitivity and CNS hyperexcitability, so that when you see a patient that has an increased pain hypersensitivity, you said, you have central sensitization, CNS hyperexcitability. And this is my problem. This is what I say, that this is a kind of gospel thing that I say. The problem with that one is that scientifically you've jumped over one step, and that is the link between the two. And the link between the two is far from being established. Nobody's measured neuronal excitability in patients, particularly after a long period of time. We don't know which systems are relevant to pain perception in animals when we study them. I mean, we study the system, we see that it increases in excitability, but we don't know which ones are relevant to pain perception. And we haven't correlated CNS excitability with sensory perception in people. And that is an important gap in this knowledge. So just as an idea, there are many other ways that we can explain, or that we can approach, not necessarily explain, that we can approach the problem of pain hypersensitivity. Vani Apkarian, a few years ago, started this study in which he showed that there are actually structural changes in the cortex and the thalamus and other aspects of the brain in patients, this is in human beings, which are related to the presence of chronic, in this case was chronic low back pain. But That was also, has been also studied in chronic conditions such as tension headache, fibromyalgia, irritable bowel syndrome, and also interstitial cystitis. So there are structural alterations in the brain in the long term that may be probably a better explanation for changes in pain hypersensitivity. And of course, because you have alterations in the structure of the brain, you also have impaired emotional decision-making tasks and impaired working memory. And you start seeing more of the symptoms that you see in the patient with chronic pain in a condition such as interstitial cystitis. So just as this is the wrap-up message, a persistent pain state might be caused by CNS alterations other than an increase in neuronal excitability. Just think about it. I mean, I know it's early in the morning, but at least I give you something to think about it. And in particular, that we in the lab, we look for basic reflexes, neurons and pathways from the bottom. Clinicians, and we find hyperexcitability, but that's where we end. Clinicians, on the other hand, look to patients and they find pain hypersensitivity. But there is, of course, a huge gap in the middle. And as they say in the London Tube, mind the gap. So thank you very much.
Do you feel that the like antidepressants and so forth has a role in chronic pelvic pain based on central effect or peripheral effect?
Well, yes, the antidepressants, particularly the inhibitors of reuptake of serotonin and so on, act mainly at the CNS level. And those some can be effective in situations of this kind of functional pain, like irritable bowel syndrome, interstitial cystitis, and so on. At that level, I think it would be a central action, pharmacological action. But on the other hand, we haven't really completely explored all the possibilities of what happens at the peripheral end, and some of the conditions that cause these functional pain syndromes in people. We can't find anything in the periphery, but that doesn't mean that there isn't anything there. So it may be something more subtle that we haven't still found. So I will keep an option open. Although when you treat a patient with antidepressants of the serotonin reuptake inhibitor type, then it would be more likely to be a CNS site of action.
I have a question. So there is evidence that there's some cellular changes, some intracellular signaling that could mediate this central sensitization. Is there any evidence of reversibility once you take away the noxious stimuli? Is there over time a reversibility or reversible changes occurring?
Well, as I said, in humans, nobody's really found central sensitization in terms of what we understand by it in the lab, which is a synaptic process and of increased excitability, particularly in the long term. In animal models, all the studies are short-lived by definition. But even those that are longer-lived, everything reverts to normal after an acute insult. So if you do any kind of acute noxious input in an animal model, you may have an increase in excitability that may last for a few days at the most, and that's it. So it's difficult to make that into a kind of chronic process. Something else has to come in order to maintain this high increase in excitability. And I think that there may be structural alterations, hormonal changes sometimes. I mean, we've done a lot of work with this estrogen, for instance, and Jeff Mokel will talk about sex differences immediately. So there may be other things which are not necessarily a very high excitable CNS for months or years. I mean, I find that difficult to comprehend myself.
Great. Is there any other questions for Dr. Cervero? Great, thank you.